excretory genital ducts or excretory passages. They are the tubules that link each testis to the urethra. So, they convey the spermatozoa and semen to the exterior. They are either within the testis intratesticular excretory tubules or outside the testis extratesticular excretory tubules. Those intratesticular excretory tubules are tubuli recti. They are the end straight tubules of the seminiferous tubules, lined by Sertoli cells. Red testis. They are branching and anastomosing tubules in the mediastinum testis, lined by cubical cells. Vasa efferentia. They are about 12 tubules arise from the mediastinum testis. They are lined with tall and short, ciliated and non-ciliated cells resting on basal lamina and is surrounded by a thin circular layer of smooth muscle fibers. Extratesticular excretory tubules. Epididymis is formed of head, body, and tail. The head is composed of the convolutions of the vasa efferentia, while the body and tail are formed of one long, highly convoluted duct. Structure. It is composed of mucosa lined by pseudostratified columnar ciliated epithelium composed of basal and principal cells. The basal cells are round and undifferentiated, apparently serving as the precursors of the columnar principal cells, while the principal cells possess stereocilia, very long microvilli. Then there is lamina propria of thin loose connective tissue. Musculosa is a circular smooth muscle layer surrounds the basement membrane of the epithelium. Adventitia is a connective tissue layer surrounding the outer surface of the duct. Vas deferens is a long, straight, thick muscular tube that begins at the tail of the epididymis in the scrotum and ascends within the spermatic cord and passes through the inguinal canal to enter the abdomen. It expands to form the ampulla near its termination, where it joins the duct of the seminal vesicle to form the ejaculatory duct, which opens in the prostatic urethra. Structure. Vas deferens is formed of mucosa of irregular, slightly folded, and is composed of epithelium, pseudostratified columnar epithelium with stereocilia, and corium of loose connective tissue. Musculosa is very thick and is the dominant features of the vas deferens. It is formed of inner and outer longitudinal layers and middle circular layer. Adventitia is made of thin, loose connective tissue. Ejaculatory duct is a short, narrow continuation of the vas deferens beyond the ampulla and joins the duct of the seminal vesicle and empties into the prostatic urethra. It is lined by pseudostratified or simple columnar epithelium, but it lacks a muscular wall. But instead, it has a layer of fibrous connective tissue. Male urethra is divided into prostatic urethra lined by transitional epithelium. Membranous urethra lined by pseudostratified colaminar epithelium. Benign or cavernous urethra lined by stratified colaminar epithelium, which changes to stratified squamous at the opening, fossa navicularis. Comparison between seminiferous tubule and duct of epididymis. Seminiferous tubule has a narrow lumen about 200 micron in diameter and is about 50 centimeter long, while duct of epididymis, on the other hand, has a wide lumen about 400 micron in diameter and it is about 6 meters long. Structure. Seminiferous tubule is lined with highly specialized stratified epithelium, spermatogenic epithelium, formed of Sertoli cells and spermatogenic cells. The epithelium is resting on a basement membrane. It has no muscle layer outside the basement membrane. Duct of epididymis, on the other hand, is formed of mucosa, musculosa, and adventitia. Mucosa is lined with pseudostratified colaminar epithelium with sterocilia, long microvilli, and it has a narrow connective tissue corium or lamina propria. Musculosa is formed of circular smooth muscle layer surrounding the basement membrane of the epithelium, and near the vas deferens it becomes composed of 3 layers, then adventitia of loose connective tissue. Function. Seminiferous tubule is the site of formation of sperms, while duct of epididymis is the site of storage and maturation of sperms, the resorption of testicular fluid, phagocytosis of residual bodies. Comparison between ureter and vas deferens. Ureter is an excretory passage for urine, while vas deferens is an excretory passage for semen. Ureter is long, has thick wall, wide and folded lumen, while vas deferens is short, has thin wall, narrow and less folded lumen. Structure. Ureter is composed of mucosa, musculosa and adventitia. Mucosa consists of transitional epithelium and a wide connective tissue corium. Musculosa is thin, formed of 2 layers in the upper part, inner longitudinal and outer circular, and is composed of 3 layers in the lower part, near the urinary bladder, inner and outer longitudinal and middle circular. Vas deferens, on the other hand, is composed of mucosa, musculosa and adventitia. Mucosa consists of pseudostratified colaminar epithelium with sterocilia and a narrow connective tissue corium. Musculosa is very thick and prominent, formed of 3 layers, inner and outer longitudinal and middle circular, then adventitia of loose connective tissue. Male accessory sex glands. They are seminal vesicles, prostate, and pulpourethral glands. Both seminal vesicles and prostate depend on testosterone for development and functional maintenance. Seminal vesicles. They are 2 highly tortuous tubes, the duct of each unites with the terminal end of the vas deferens to form the ejaculatory duct. Seminal vesicle is formed of mucosa. It is highly folded and lined by pseudostratified columnar secretory epithelium or tall columnar epithelium surrounded by loose connective tissue corium. Musculosa is made of inner circular and outer longitudinal layers of smooth muscle fibers. Then adventitia is made of thin fibroelastic connective tissue surrounding the seminal vesicles. Functions. The secretion of the seminal vesicles is thick, yellowish, alkaline, fluid, rich in protein, fructose, and vitamin C. These are important for nutrition and production of energy for sperms. Prostate is an example of male accessory sex glands. It is easily diagnosed by the naked eye and eyepiece. It is a composite gland formed of 30 to 50 compound tubuloalveolar glands surrounding prostatic urethra, from which numerous ducts drain independently into the prostatic urethra. The gland is made of stroma and parenchyma. The stroma is made of capsule and trabeculae formed of fibromuscular connective tissue, rich in smooth muscle fibers. The stroma surrounds the urethra, where the muscle fibers form the internal sphincter of the bladder. Parenchyma is arranged concentrically in 3 levels around the prostatic urethra. Mucosal groups around the urethra, submucosal groups outside the mucosal glands, outer or main groups found underneath the capsule, where their acini are larger than those of the previous groups. The acini and ducts of the prostate are lined with simple columnar epithelium. The glandular epithelium differs greatly from gland to gland, and even within the single alveolus. It is usually simple or pseudostratified columnar, but may be low cuboidal or squamous. Functions. Prostate secretes a thin, milky, alkaline secretion which gives semen its characteristic smell and neutralizes the vaginal acidity. Secretion is rich in acid phosphatase. Clinical notes on prostate. Mucosal and submucosal glands enlarge after the age of 40, causing pressure on the urethra and difficulty in micturition, a condition known as senile prostate. On the other hand, carcinoma of the prostate affects the outer glands. It is diagnosed by the presence of high levels of acid phosphatase in the plasma. The penis. It is the copulatory organ by which semen can be transferred to the female genital tract. By eyepiece, it is made of 3 corpora, 2 dorsal corpora cavernosa and 1 ventral corpus spongiosum, which expands anteriorly into the glans penis. Each corpora is made of erectile tissue, which consists of blood spaces separated by connective tissue and smooth muscle fibers. The corpora are richly supplied from branches of the dorsal artery of the penis and surrounded by a thick layer of dense connective tissue, which is called tunica albuginea. The three corpora are surrounded by another layer of connective tissue, the fascia penis. The two corpora cavernosa are separated by incomplete septum, which allows free communication between the two. The corpus spongiosum, on the other hand, is traversed by the penile urethra, which is lined with stratified columnar epithelium. The skin covering the penis is thin and is free from subcutaneous fat.
بسم الله الرحمن الرحيم يسبح لله ما في السماوات وما في الأرض له الملك وله الحمد وهو على كل شيء قدير